All right, thanks everybody for having me. Just as an update, I've been tasked with discussing systemic therapies and clinical trials, but I am not going to be able to do justice to all of the research work that's being done. So I had to focus on a couple of different areas that we're looking at. But there is a lot more. And if there's any questions that people have, happy to discuss in greater detail. So this is only a subset of the full slide deck that I would have liked to present here today. Um, So I wanted to go a little bit into trying to understand the basic science of desmoid tumors, right? So we know the way that I try to explain desmoid tumors to patients that I see in clinic is that these are mutant scars. I think a lot of people have obviously familiarity with scar tissue, normal scarring, that entire process, how if you injure yourself, you'll have a scar, a bump will form, and eventually over time that will regress and flatten out. Some people are also familiar with the diagnosis of a keloid. Some people will know, for example, after a piercing, they'll know a friend who actually developed a bump that kind of increased in size and became fairly significant. Well, desmoid is the extreme, right? So oftentimes when I think about it, it's a mutant scar. And so that the cells that are actually the ones that are actually, let me see, whoop, that did not work. So the cells that are actually forming those scars are the myofibroblasts. So those are kind of cells within the body that actually create collagen, create those scar tissue, help the body to heal. And so there's a very, very carefully coordinated series of events that happen within that cell to help it actually heal tissue. So let me see here. So what makes desmoid tumors different? What makes them tick? What do we understand about the basic biology? So um, we know that there are mutations that occur in these myofibroblasts, right? So what causes those mutations? There's still a lot of active research in terms of those questions, but these are not healthy, normal myofibroblasts, right? So if you mutate a myofibroblast, you get a desmoid cell. So that's exactly what they look like. I think our pathologist can tell us exactly under the microscope. You know a desmoid when you're looking at it. So what are the mutations that we see? I think over the last 20 years, there's been an increased understanding of the specific genetic changes that we see in desmoid cells. So with the advent of genomic technology and genomic sequencing, they've done a fairly broad look at the potential genetic changes that you see in desmoid. And most patients have a very, very specific set of mutations in a very, very specific gene called beta-catenin. So 20,000 genes, almost everybody with a desmoid, if you look at that desmoid cell, will have a mutation in that beta-catenin gene. We say about 80 to 85%. And most of these are not inherited, right? So the vast majority of these beta-catenin mutations will occur only within those cells. So some event happened to mutate those. You can see there's a slightly higher rate of beta-catenin mutations in patients as we get older, slightly lower in patients who are younger. And then if we look specifically, the most common mutations are given certain names. As Dr. Tinoco said, there's not going to be a quiz on them, but T41A, S45F. There's been a lot of research in terms of what is the significance of these mutations. What do we know about these mutations? It makes the beta-catenin very hard for your body to break down. So it starts to build up within cells. And so the more beta-catenin you have in cell, the more actually the cell will grow. So I put this diagram up just to kind of make sure that everybody's going to see this diagram. There's not going to be a quiz about it. Half of us from physicians probably would mislabel all of those specific genes in that particular pathway. But just think about the signaling. How does a scar tissue cell know that it needs to heal a wound? What's going on? The nucleus or the center of that cell is exactly where that growth is controlled and where that programming of that cell takes place. And we know that beta-catenin actually has to work its way into the center of the cell in order for it to actually activate those growth pathways. And so there's essentially a game of telephone, right? So signals will come at the cell surface, they'll bind to the cell, and essentially a game of telephone. The proteins on the surface will tell the genes that are closer to the surface that activation needs to happen. And eventually that will lead to the beta-catenin working its way into the nucleus. As Dr. Tinoco said, we most of the time beta-catenin itself is mutated, but you can have mutations in any of those other parts of that signaling cascade, potentially to also lead to beta-catenin entering into the nucleus. So as we see here, as beta-catenin works its way into the nucleus, it will interact with a couple of different proteins within the center of that cell. And so people have wondered for some time, are those druggable? Is there something that we can actually do to actually target that interaction between this key molecule? So this is kind of, once again, my understanding of kind of how desmoids work, right? So essentially you have this carefully choreographed control system, and essentially your switch is broken. So it's stuck in the on position. You just keep piling up these mutant cells, right? They think they're doing their job, and they're not actually needed. So I had to limit in terms of kind of what we're going to talk about today, and I chose to focus on kind of some research that we've been part of at Ohio State, other research that's happening here in the state of Ohio that we're not currently part of, all focused on this particular pathway. So the first one is actually looking at that beta-catenin interaction with a gene called TBL1, right? So this is actually within the center part of the nucleus. So beta-catenin can't drive cell growth unless it actually binds to that TBL1. And so we were part of some very, very early work. Dr. Pollack's lab here investigating within desmoids, how does this pathway work and how do these drugs actually interact with that pathway? Then I'll kind of touch a little bit later on a compound called OG01, which is actually targeting a slightly different part of that pathway. So this is a little bit of a cartoon. You can see here that in this cartoon, tegavivint will actually bind to this TBL1. Beta-catenin, when it's actually in the nucleus of the cell, has to bind to TBL1 to be stabilized, right? And so when it actually doesn't bind to that TBL1 partner, it can be degraded. So this was kind of the rationale for that particular concept. And so this is a slightly different version of that same concept, right? So if beta-catenin can't be stabilized by TBL1, beta-catenin can actually be more effectively broken down, right? So can't signal, growth will be turned off. And essentially, as we talked about, this is a mutant scar. If it's not constantly producing new myofibroblasts, not constantly producing new scar tissue, in theory, the healing process can actually finally kick in, and the cell can actually, the desmoid can actually start to regress. So we were part of the study. Dr. Tinoco has kind of gone over these particular waterfall plots. So once again, the more bars going down, the better things are. I will point out, in this study, the overall response rate to tegavivint was 17%. And this was in a number of heavily pretreated patients at that point in time. So many patients had received multiple prior lines of therapy, essentially looking at a very novel therapy. This is what we call a phase 1 study, where we're looking to figure out what the dose of the drug actually is at this point in time. So each color bar was a slightly different dose. They moved the doses up and tracking side effects, and overall 25% of individuals responded at a recommended dose, at the recommended phase 2 dose. Certainly not the response rate that we were hoping for at that point in time, but it's certainly to give us an early signal that these direct beta-catenin targeting agents do have activity in desmoid. I don't talk about it here, but I do also want to kind of point out that this is the FDA's preferred definition of response rate. A lot of these patients who may not have had shrinkage still did demonstrate symptomatic benefit. So oftentimes we'll see changes in terms of blood flow and so forth that results in improved symptoms. And so there are other ways to look at response rates, but at least per the standard drug development definition of what constitutes a response, we're looking at about 17 to 25% of patients. So this was kind of exactly the same slide. You'll see these a lot here today. We know that TBL1 is important in terms of that stabilization of beta-catenin, but once again, it doesn't actually do its job until it binds to another protein within the nucleus, so TCF. And so there's been a lot of interest as well in developing kind of newer therapies, and so this is actually a drug under investigation. Trial is actually open in Ohio, in Cleveland at this point in time. Fortunately, we don't have it open at Ohio State. As I said, we do partner with everybody within the region in terms of finding these studies for patients. This is actually a very exciting study as well. So they've presented their data. We've actually referred to Cleveland for folks as well. So this is in the first 10 patients that were reported in this study, there was an 80% objective response rate in patients who had actually had at least more than one baseline scan. And so of these patients, several of them were heavily pretreated. Most patients had seen multiple prior lines of therapy, between 1 to 2 prior lines. And at least within this very, very small subset of patients, there was no evidence that it was preferentially working in patients with a certain mutation, certain location, certain prior therapies, or so forth. So a very interesting study. And so this is the data from their initial presentation. This is the standard waterfall plot. Just keep in mind, more down bars indicates a better overall response rate. And so of the patients who had actually had multiple prior scans in this study, 80% actually achieved that response rate. So very exciting stuff. Looking forward to kind of hopefully seeing this move forward. So especially in patients where kind of standard therapies don't work. And so this is actually just kind of just to highlight some of the responses seen in this particular study. Here. So we can see in some of those areas where we actually have pretty significant decrease in, once again, I've said that glow, that brightness, that bright white contrast enhancement in these patients. So very, very encouraging in particular in that study. So the last thing I wanted to talk about here today was, oftentimes, we think about these, at least in the research that I chose to present here, looking at that beta-catenin mutation, that beta-catenin pathway, how does beta-catenin drive growth? Just keeping in mind that there's a lot we still don't understand, right? So spontaneous regression, we don't do anything and the tumor spontaneously resolves. Similarly, drugs like sorafenib do not target that beta-catenin pathway. I think the folks, in terms of the nirogacestat, which is also approved, and the other gamma secretase inhibitors, we have some thoughts about how they might interact with the beta-catenin pathway, but do we honestly know if that's the primary mechanism? We don't really know. And so there's a lot of work that still needs to be done in terms of kind of how do these tumors respond and how do they, just tracking response. And so there's a lot of work here that I'm not going to go into, but I did want to get just one taste of kind of looking at outside the cell and looking at kind of some of what we call the stroma, right? So we know that the tumor cells are releasing a lot of factors into the environment, right? So there are trials looking at what are they releasing, TGF beta, and there are some interesting studies in terms of like tracking desmoids, right? So how do we understand if a desmoid is actually active, right? So some studies have looked to say, is there something that we could be looking at in the stroma to try to understand a little bit better what's going on? So I mentioned this. This is I know Dr. Tinoco and I actually, this is an idea that we had maybe about 8 years ago, right? Where we were actually working with some of the folks in terms of tracking different types of proteins that were present actually in the stroma. So we know that desmoids produce a lot of protein, right, that they'll release. And so a lot of a desmoid is the cells as well as the protein. And so there are high levels of certain proteins. One particular called fibroblast associated protein, FAP. This is not the same FAP as we've been talking about before. So this is a protein specifically. Brown indicates high levels of expression. So here's a desmoid tumor, very, very high levels of FAP. It's actually among the highest levels that we'll see in any tumor, and almost none in kind of adjacent healthy tissue for these patients. So actually, there are scans that can actually measure how much FAP is present in a tumor. So this is actually a scan of a patient actually with a mesenteric desmoid. So this is a very specific study where they actually give a radioactive molecule, which actually binds specifically to FAP, and you can actually identify where the FAP is actually active in that patient. So this was actually a study that came out of Germany, which is kind of one of our early partners kind of in this entire process, looking at desmoid tumors, and they've tracked this among a number of different patients in terms of seeing how do these tumors respond, right, at baseline, kind of following initial progression. And then actually, as the patient responded to therapy, you'll see a little bit less glow in that patient. There's a lot of ideas that people have. Some people are kind of looking at kind of pairing kind of drugs that target this, radioactive molecules that can target this as well. So this is an exciting area, I think, at least, in terms of tracking it. And so, like I said, this may not be relevant for desmoids, but we certainly are using kind of some molecules that can actually deliver small doses of radiation to the areas that light up on those scans. So we don't use radiation nearly as much as we did in the past for desmoid, but this is a very highly targeted form of radiation. So this is an area of active research. So, and with that, I'd like to thank you guys for including me. I couldn't cover everything that we would have liked to, but if you guys have any questions at the end, happy to field them. So.